Human Immunity by Hollie, Wheaton, Unit 17. Within my presentation, I am going to be speaking about the first, second and third lines of defence and what they mean, the third line of defence and the roles of different types of immune cells and immunoglobulins, self-recognition and tolerance to anti-tissues and rejection of transplanted organs, two specific immune system disorders, the categories of acquired immunity including natural, artificial, passive and active, the immune response to vaccination including immunological memory, the benefits of vaccination, analysing measles infection rates in the UK between 1940 to present, and lastly herd immunity, vaccination programmes and factors affecting vaccine uptake. The first line of defence prevents pathogens from entering the body through physical and chemical barriers. Some examples are the skin. It protects the body from pathogens. Once the skin is damaged, it starts to repair itself by forming a scab, which is a physical barrier. The eyes produce tears, which contain enzymes to protect the eye, which is the chemical barrier. The nostrils contain hairs, which is a physical barrier. This prevents pathogens from entering. The nose contains mucus, which traps the pathogens to prevent them entering towards the lungs. The respiratory tract contains thin hairs that sweep the pathogens away from the lungs and out towards the esophagus. This is known as cilia. When a person coughs or sneezes, they are removing the pathogen from their body, which is a physical barrier. The digestive tract contains mucous membranes, which trap the pathogens. Mucous membranes are found within the nose, trachea, mouth, lungs and stomach, which is also So the first lines of defence, here we have 6 examples. We have saliva, which contains antibacterial enzymes. We have the skin, which prevents entry. We have stomach acid, so low pH levels, which kill harmful microbes. We have tears, which carry antibacterial enzymes, the same as saliva. We have mucous linings, which trap dirt and microbes in the esophagus. And we have good gut bacteria outcompete bad in the stomach. What is the second line of defence? So the second line of defence occurs when a pathogen has entered through the first line of defence. For example, a cut on the skin which has become infected. So once a pathogen has entered through the first line of defence, chemical signals are released to recognise this, such as histamine, which is a hormone, which helps blood vessels to widen to enhance blood supply and results with its inflammation. Signs of inflammation are redness and warmth around the area. The blood around the infected area has increased in temperature to destroy the infection. Macrophages. So macrophages are white blood cells which assist to destroy pathogens in the body. The macrophage attacks foreign bodies to eliminate and prevent infection. Macrophages circulate within almost every tissue in search for pathogens to eliminate dead cells. The table on the next slide describes the locations and the functions of macrophages. So here we have 4 types of macrophages within the locations and functions. So firstly, alveolar macrophage, located in the lung alveoli. Its function is phagocytosis of small particles, dead cells or bacteria, initiation and control of immunity to respiratory pathogens. We have Kupffer cells located in the liver, which initiate immune responses and hepatic tissue remodeling. We have the microglia within the central nervous system, and its function eliminates old and dead neurons and controls immunity in the brain. And we have splenic macrophages within the spleen marginal zone, red and white pulp, and it eliminates dysfunctional and old red blood cells. So what is the third line of defence? So the third line of defence is the third and final line and occurs when the pathogen bypasses the first and second line of defence. The immune system is needed to achieve this. Types of pathogens include viruses, bacteria, fungi and parasites. There are 2 parts of the immune system, including innate and adaptive, which collaborate together to kill the toxins. Macrophages and dendrite cells kill the pathogens. This is known as phagocytosis. The pathogens are then broken down into small parts which enter the major histocompatibility complex family, also known as MHC in short. The MHC proteins line around the cell's surface, which in turn causes the antigen-presenting cells, also known as APC in short, to travel to the lymph node. Lymph nodes are all over the body, such as T cells and B cells, which are found in our lymph nodes, which originate from the adaptive immune system and are also known as lymphocytes. So acquired immunity. There are 2 types of acquired immunity within the immune system, which are active and passive. They both contain natural and artificial antibodies. So naturally active acquired immunity is a natural process of an infection. The body creates antigens to protect against infection. Naturally active acquired immunity contains memory cells. Once the body is exposed to the antigen, or if exposed to the same antigen for the second time, the body creates antibodies quicker as it has already been exposed to the toxin. Thus, immunological memory is created to recognise this. Artificial active acquired immunity is when a person is exposed to a foreign antigen with intent of revaccination, which in turn causes the body to create antibodies. Natural passive active acquired immunity does not contain memory or plasma cells. There are two types, which include placental transfer of IgG from mother to baby during pregnancy. Once baby is born, the antibodies last from birth until 6 months. The second type is found in colostrum, which is mother's breast milk, which contains antibodies to protect the baby after birth. Artificial acquired passive immunity is when antibodies from another person are either injected or transfused into another person's body. So here, as spoke about previously, we spoke about acquired immunity. So here we have a little diagram just explaining more in detail. So the blue box, acquired immunity, which is immunity that develops during your lifetime. Over to the left in the green box is active immunity, which develops in response to an infection or vaccination. So the natural side are antibodies developed in response to infection, and the artificial side of active immunity is when antibodies are developed in response to a vaccination. Moving over to the right in the orange box, we have passive immunity, which develops after you receive antibodies from someone or somewhere else. So the natural side of passive immunity is antibodies received from mother, for example through breast milk or through the placenta. And we have the artificial side where antibodies received from medicine, for example through a gamma globulin injection or infusion. Immune system cells contain leukocytes, which are white blood cells within the immune system. White blood cells defend against antigens and infections. There are 5 different types of leukocytes, which are produced in bone marrow from multipotent cells, also described as hematopoietic stem cells. White blood cells are found all around the body, such as connective tissues, bloodstream and lymph system. The table on the next slide explains a bit more in detail the 5 types of white blood cells. So here we have 5 types of white blood cells. We have the neutrophil, which is multi-lobed and its function is bacterial or fungal infection. They are the most common first responders to microbial infection. We have the examples on the right just so we know what they look like. We have the eosinophil, which is bilobed, and its function is parasitic infections and allergic reactions with an inflammatory. We have the basophil, which is bitrilobed, allergic and antigen response, which releases histamine, causing vasodilation. We have the lymphocyte, which is deep staining eccentric. Its function is include B cells, CD4 and helper T cells, and CD8 and cytotoxic T cells. Operate primarily in the lymphatic system. We have the monotype, nucleus is kidney-shaped, and the function is phagocytosis of pathogens, presentation of antigens to T cells. Eventually they become tissue macrophages, which remove dead cell debris and attack microorganisms. Here we have immunoglobulins. So immunoglobulins are proteins which are found in the blood, which is also known as plasma, which creates antibodies from bone marrow. Immunoglobulins travel along the bloodstream to kill off pathogens. If the body doesn't produce enough antibodies, the body starts to deteriorate by causing an infection. There are 5 types of immunoglobulins, including IgA, IgD, IgE, IgG, and IgM. Ig stands for immunoglobulin. Immunoglobulins are made up of 2 light chains and 2 heavy chains. The 2 light chains are known as kappa and lambda. There are 5 types of heavy chains such as A, D, E, G and M. And on the right here we have a little diagram just explaining about the light chains, the heavy chains, the immunoglobulins as well as the plasma cell. So types and functions of immunoglobulins I spoke about previously in the previous slides. Here we just have a table explaining a bit more in detail. Recognising self and non-self antigens. So every cell in the body contains antigens which have proteins, nucleic acids, fats and carbohydrates. The antigens are recognised as self-antigens, however, cells that do not originate from the body are recognised as non-self antigens. Self-antigens are inside all cells within the body and reside in blood cells. For example, a person who is undergoing a blood transfusion can only receive the transfusion if the donor has the same type of antigen. Otherwise, the immune system will attack the donated blood, as the immune system will not recognise the blood cells as being self-antigens. Non-self antigens occur within bacteria and viruses and are also present in blood and organ transplants from a person who has different antigens to another person. If there are non-self antigens within the body, the immune system will destroy them. Lymphocytes, also known as white blood cells, recognise and react to non-self antigens. MHC1 and MHC2. So here we have a table with two columns just explaining more about MHC class 1 and MHC class 2. Rejection to transplanted organs now. So an organ transplant is a replacement of damaged organ to a healthy organ. Organs are things such as skin, liver, heart, lungs, eyes and so on. Transplants save lives and allow people to live for longer. A donated organ can come from a donor who is deceased. This means that they gave their consent before their death, or from a living donor who donates things such as the kidneys. A person can still have a healthy life with only having one. In order for successful transplantation, the donor and the patient will have to share the same blood group, or the transplant will reject against the patient. Organ transplants can reject due to a person's immune system thinking it's a foreign body and therefore destroys the transplant tissues, similarly to when the immune system kills off pathogens. In order to prevent organ failure, the patient is given immunosuppressant medication. However, this can sometimes lead to infections and failure if the patient is not given the correct amounts. Transplant rejection. So here we have three types of transplant rejection. We have the hyperacute, we have the acute, and we have chronic. On the right, there are some common signs and symptoms of transplant rejection also. Two types of autoimmune diseases. So we have lupus, which is a type of arthritis, where the body attacks itself by cells attacking the internal organs within the immune system. There are four types of lupus, however the most common one is systematic lupus, which attacks organs, joints and skin. Lupus is not contagious, therefore a person will have to have the genetic fault to have this type of autoimmune disease. We also have type 1 diabetes, which occurs in children, however it can occur in adults. The immune system attacks the insulin, which is a natural hormone. Insulin comes from the pancreas, which travels through the bloodstream and into cell tissues. Without enough insulin, glucose levels pile up within the bloodstream, causing high blood pressure. Type 1 diabetes can lead to heart attacks, strokes and limb amputation due to damaged tissues and bone. There are treatments to control this condition, such as insulin injections, which are injected into the side of the leg or the stomach, and insulin pumps. Cannulas are inserted under the skin and are in the bloodstream. This is normally placed near the waist. So here we have some symptoms of systematic lupus. And here we have some symptoms of type 1 diabetes. Immune response to vaccination and immunological memory. So vaccinations are injected into the muscle of a person, preferably the arm. Vaccines contain live and weakened pathogens to encourage the immune system to strengthen and protect itself from the diseases in the future. This is known as the first response. Once someone is exposed to the pathogen again, the immune system will defend against illness, as memory cells from the vaccine will remember the pathogen and will create antibodies faster. Leukocytes stimulate during primary exposure of a pathogen. Once the pathogen has been destroyed by the white blood cells, the cells remain within the immune system until the individual is re-exposed again. Immunological memory occurs after the first primary response against vaccination and protects an individual from future infections. Immune cells such as antibodies, B cells, T cells, CD8 cells and CD4 cells collaborate to achieve this. This can be achieved by vaccination, natural infection or therapeutic interventions which introduce the body to a specific antigen which then initiates the immune response. So the benefits of vaccination. So immunisations are given to children to protect from diseases, build and strengthen their immune system to future diseases that can cause harm. It also protects children from catching diseases off of other people around them by creating antibodies. During the 1900s, infectious diseases were at an all-time high, causing death. However, vaccinations now have prevented many infection and death rates in the UK. There can be some side effects from a vaccine, however, this is very common and not serious. Some of the most common side effects include a red area around the injection site, which can feel sore, swollen and itchy to the touch, Babies and children can feel unwell, so they may have a high temperature, gone off of their food, and tiredness. Allergic reactions from vaccinations are very rare. However, if an allergic reaction arises, which is also known as an anaphylactic shock, this can be treated quickly with treatment. Vaccinations contain a bacteria or a virus which is either live or killed within a laboratory. A person cannot catch or spread a disease from any vaccination given to them, as it has been weakened, and it is purely to build, strengthen and protect the immune system from future diseases. So here we have vaccinations given throughout a person's lifetime. We have a little table, so it's from childhood up until adult age. Measles infection rates between 1940 to present. So on the left here we have a diagram of measles rates. Herd immunity. So herd immunity is a theory around vaccination. Once a certain amount of the population has become infected and immune, a virus slows down and does not increase the amount of infection rates within the population. This means that most people are already immune and thus prevents infection rates from rising and reduces infections from spreading. Once a vaccination is given for a type of infectious disease, herd immunity will become protected for future outbreaks. For example, the Spanish flu, which originated in 1918 to 1920, was a global pandemic that died out, as well as swine flu, which occurred in 2009 to 2010. In order to reach herd immunity, there are two ways, which include natural infection, which is a global pandemic, or vaccinations to build immunity and prevent future outbreaks by reducing the disease. Vaccinations are given to the population to prevent disease and death. It achieves herd immunity. Vaccines are given to children and adults. However, some people can be exempt from some vaccinations with reasons such as if they have an illness which weakens their immune system because of the vaccine, if they are allergic to ingredients in the vaccine, if they have had a previous allergic reaction to the vaccine, religious beliefs, personal views and beliefs. Some people have complained that the MMR, also stands for measles, mumps and rubella vaccine, causes autism in children. In order to qualify as medically exempt, a doctor will need to sign an exemption form as proof. Factors affecting vaccine uptake. So here we have another diagram. So vaccine hesitancy, we have social factors, we have cultural factors, demographic factors, psychosocial factors within vaccine uptake. So within this presentation, I have spoken about non-specific defences against disease by describing the first and second lines of defence by giving specific examples. The third line of defence, such as acquired immunity, sensitivity to antigens and the roles of different types of immune cells and immunoglobulins. Self-recognition and tolerance to own tissues and rejection of transplanted organs, two specific immune system disorders including lupus and type 1 diabetes, the categories of acquired immunity including natural, artificial, passive and active, the immune response to vaccination including immunological memory, the benefits of vaccination, measles rates and in the UK between 1940 to present, and lastly herd immunity, vaccination programmes and factors affecting vaccine uptake.